Well, you hear me? Well, hello everyone. It's a pleasure to me to be here. Um, as you can see, my presentation is about the Pleurothallidinae inflorescence. This topic really caught my attention when I was studying the genus Myoxanthus for my master thesis a few years ago. And when I was studying this genus, I realized that there was some incongruences between morphology and how the inflorescences of the species have been described. So I decided to study this. I think that the subtribe doesn't need a large introduction since you are very familiar with, but it's a mega diverse group of orchids, and there is more than 5,000 species found in the neotropics. And just in the last year, 83 new species were described for this group of orchids. Um, well, when I was studying, oh, sorry. First of all, I think that it's very important to make a review of the general morphology of these orchids. First, we have sympodial growth. We have a sympodial unit here, which always has a basal structure, which produce roots, and then it has aerial stem, which with a apical but not terminal leaf, and the terminal shoot produces the inflorescence, as you can see here at the apex. So what is typology? It refers to the study of the classification the different types of the flowers arrangements on the plant. Based on these arrangements, can be we can find different classification of the inflorescence. So it is true that the inflorescence, its morphology, and the size of the inflorescence, the different aspects of the inflorescence, um, affects the general appearance of the plants. But it is also a fundamental criteria for establishing relationships between plants. And however, it is only true if we are starting homologous structures. As you can see here, it is extremely relevant when comparing taxa because superficially appearances may not result from homologous typologies. When I was preparing this presentation, I was trying to find a species of Pleurothallidinae that represents very well the inflorescence of the group. So after thinking and thinking, I decided to use this species. So apparently you're thinking this guy don't even know how to recognize a Pleurothallidinae species, but don't worry, I do. So I know that this is an Oncidium species, which belongs to another subtribe. But then you will see that this is a very good example to represent a Pleurothallidinae inflorescence. So we are going to study a Pleurothallidinae species, the inflorescence of Pleurothallidinae species. So let's address a Pleurothallidinae inflorescence of Pleurothallidinae species. Here we have this Anciathera species. So first we are going to remove the spade at the base of the inflorescence. Then we are going to cut the leaf, and then we're going to remove all these bracts at the base of the inflorescence. So we got something like this. What you can see here is the apex of the ramical without the spade and without the leaf, only the inflorescence. As you can see at the base, we got something like a very compact branch system at the base of the inflorescence. So, and this is not the inflorescence of the species. This is just a part of the inflorescence of the species. This is whole the inflorescence of the species. So this is a suppression of internodes at the base of the inflorescence, which results in a very compact branch system in Pleurothallidinae. After studying a lot of material or living material of this subtribe, it seems that this is a common feature of the subtribe. As you can see here, just the base of the inflorescence, there is always a compact branch system at the base of the inflorescence. And you can see here the white arrows indicate the scar or the node where the spade was. So the internode above and below the space represent the peduncle of the inflorescence. So the peduncle is actually completely reduced in this subtribe. As you can imagine, some of them are very easily to study the typology, as some of these species of Pleurothallidinae. Here, there are just few internodes and just few branches, but as soon as we add to the branches, there are more branches and branches and branches, it gets more complicated to study. So, well, actually, a nightmare to study this type of topology. So let's take another example here. This is a species of the genus Myoxanthus. This is Myoxanthus hirsutitalis. And here we are going to study its typology. If we try to study the typology of this species, we got something like this. And then we are going just to elongate the internodes of this inflorescence. So we will get something like this, just expanding the internodes. So now, not much different now, right? Well, I know that the terminology inflorescence is quite confusing and quite different because there is a lot of terminology and classification, but we have to deal with when we have this type of complexity on the inflorescence. Following the classification of Troll, which is an author that has studied inflorescence and morphology of plants, here we have this branch system of an inflorescence, which is also called a synflorescence. Then we have the apical unit, which is called main fluorescence. And then every lateral unit is called coflorescence. And then we may have coflorescence of first order, coflorescence of second order, and so on. As you can see here, this is very similar to what we have in Pleurothallidinae. So what we are actually seeing in Pleurothallidinae is a lot of coflorescence. Now we are going to study another 3 typologies of 3 different species. First, we have here this species of Myoxanthus, this species of Pleurothallidinae, and this species of Platystellium velata. So we study this typology, we get something like this. So as a reference for you, here I show you where the spade of the inflorescence is. And as you can see, by following the position of the spade, you can see that they have completely different typologies. So that was something that I found, that the structure currently described as peduncle have been erroneously applied to this subtribe. So we are going to point out the structures that have been currently described as peduncles. As you can see here, this one is below the spade, this one is above the spade, and this one is also above the spade, so they are not homologous structures. So let's indicate the true peduncles based on those typologies. As you can see here, this one is correct, but this style of typology where the true peduncle is elongated is uncommon in the subtribe. There is only found in 3 species of the whole subtribe. There are some species of South America. Then we have in this species of Platystellium velata, the peduncle is here at the base, and the other species of Myoxanthus, also the peduncle is here at the base. And this is what is happening in most of the Pleurothallidinae. So what about the structure that have been currently described in Pleurothallidinae? Which should be the name, how have been described? Well, have been described as a peduncle, but it is actually not a peduncle. So in 2021, we decide to propose the term pseudopeduncle to refer to this non-flowering section, which of any type of coflorescence connecting the pedicel here in single flower coflorescence, so they only having one flower, or the rachis in multi-flower coflorescence, to the main fluorescence or to any other branches that you can see here, because we can have different orders. So what we are actually seeing here is a lot of peduncles, a lot of coflorescence with pseudopeduncles. And also here, we are seeing here coflorescence, one coflorescence and one coflorescence, and they are just part of an inflorescence, which is a highly compressed internal branch system at the base of the inflorescence. So after studying a lot of material, we found that there is two main types of inflorescence on Pleurothallidinae. One of them having coflorescence, producing only one flower. As you can see here, each black arrow indicates the remainder of the axis of each coflorescence or each branch. So as you can see here, each coflorescence produces only one flower. And there are just few examples of what we can find in the subtribe. There are different examples based on how long is the peduncle or based in the succession of the flowers or how they appear on the system branch. Here, you have here some examples. And the second type of inflorescence is the multi-flower coflorescence. Each coflorescence can produce 2 or more flowers per coflorescence, but they can produce a lot of coflorescence, but all of they are the multi-flower type. So there are another examples based on how long is the pseudopeduncle and based of the sequence of the flower. For example, this typology of is frequently seen in this group of Speclinia. So about the sequence, it is important. There is so many important things to talk about here, because if we study carefully the typology of some species, we may find that some multi-flower types may appear to produce single flower coflorescence. As you can see here, there's 2 species of Pleurothallidinae. They looks like producing one single flower coflorescence, but they are actually multi-flower coflorescence. Then we have this species of Speclinia producing also multi-flower coflorescence. Another important thing to mention here is the sequence and how long it takes the inflorescence to produce another coflorescence, because as you know, many species of Malvalia have been described as producing one single flower inflorescence, which is not the same to producing single flower inflorescence with single flower coflorescence, because an inflorescence with single flower coflorescence is always a multi-flower inflorescence. So these species of Malvalia have been described as having only an inflorescence of one flower, but they actually produce multiple flowers, but the next bud takes a lot of time to be developed. So here you can see the bud of the next single flower coflorescence. So they are described always as having single flower inflorescence. I know that many of you are probably thinking about the effect of these characteristics on the reproductive biology of the plants. As you know, in most, but not all, the sympodial orchids, it is common that the sympodial unit dies after producing flowers. So the sympodial unit produces its flowers and then dies, while another sympodial unit is produced to continue the cycle again. So for example, an experiment carried on the Lancaster Botanical Garden, we found, for example, that this species of Lepanthes failed to offer an upper flower just 3 to 16 days out of almost 300 days. So the species display one flower almost for the whole year. So, in the other hand, we have this example of a Myoxanthus. This species of Myoxanthus blooms only for 1 week in the whole year, but the plant can produce up to 1,000 flowers in the whole plant. It's a lot of flowers, and each inflorescence can produce up to 40 flowers per ramical. This type of inflorescence have been called perennial inflorescence because the plant is able to use the same inflorescence to continually producing flowers over several years instead of producing a new sympodial units, and this is suggested to be a mechanism to reduce energy consumption in inflorescence, in the plant. So I know that I have more time, so just in case. So just there you have a general review of their generic relationships within Pleurothallidinae. So it's interesting that in some clades, there is present just one type of inflorescence. As you can see here, this, it's too small the letters, but it represents the Restrepia affinity, where we have Restrepia, Myoxanthus, Pleurothalopsis, Echinocephala, and all those genera. But in other genera, as you can see, for example, in the affinity of Lepanthes, the multiflower type is which dominate there. But in other groups, for example, Pleurothallis, Speclinia, we can find the both type of inflorescence. More time. So, as you know, the orchid's inflorescences are of the indeterminate type. It means that at the end of the inflorescence there is never a terminal flower. Flowers are always lateral, and always you can take whatever species you want of orchids, and at the apex of the inflorescence you always will see an structure in a form of a vestigial bud, a residual, or a residual of the remainder of the axis. You can see here on a dendrobium, a variante, which is also the type of structure we find in Cattleya, and also here are the pseudoterminal flower found in Phalaenopsis. So in Pleurothallidinae, this is the remainder of the axis. I know probably some of you have seen this in the inflorescence of Pleurothallidinae. It's very, very small, and it's found at the apex of the inflorescence in the different shapes. Sometimes this is a characteristic of this vestigial structure, that sometimes this structure is fused to the pedicel. As you can see here, as you can see in the same images, these cells belongs to the vestigial structure, and though this is the pedicel. So just another examples of characteristics of the Pleurothallidinae inflorescences, which are rare on the subtribe. This is some species that is a very uncommon condition in the subtribe, where the leaf is fused to the ramical. As you can see here, all this part of the ramical is an internode, but the leaf is fused to the ramical, which the term is called coflorescence. And there is another characteristic of the Pleurothallidinae inflorescences is how they can make shifts to change. How can change the meristems to produce a vegetative structure instead of a floral structure. As you can see here, this is a way to produce more plants even from the same plant. So there are clones, as you can see here in this species of Pleurothallis, with a lot of roots here. There are a lot of other plants containing the same genetic material. So, and that's it. Thank you.
We have time for the questions.
Yeah, Thanks for your wonderful presentation. Can you show us about the in-process patterns onto the backbone phylogeny?
Sorry?
You compared the in-process types on the backbone phylogeny tree.
This one.
Yeah. From this phylogeny tree, explained in-process shows very convergent evolution. The result of the convergent evolution. Then how can you suggest pleasure-morpho apomorpho states in this clade?
Sorry, I didn't understand.
Look at that. The types of processes are very mixed one at every clade. Then if the fluorescence shows the result of evolutionary trends, there should be some possible hypothesis from where to where. But at every clade, it is mixed up. Then that's why we call this kind of thing as convergent evolution or parallelism.
Yeah.
What do you think about that?
Well, I think that this is, I actually don't know. Well, but this is true that in some clades there is one type of inflorescence. There is just as in other questions done in other talks, there are some things that we cannot explain. For example, in some groups, the vestigial structure of some inflorescence were completely lost. So I think that actually I don't know. It's a very good question. So
Gustavo, beautiful presentation. Did you find something similar like profile or cataphil in this structure in the architecture of this inflorescence?
well, this is just different terminology for some of the structures found in inflorescence. So there is there is those terms as bracts. So there is some terms of profiles. Certain those terms as sheets. So there is some terms of there is different terms, but I think that there is not like terminology on the different foliar structures found in the inflorescence. All what we found in the inflorescence are just small bracts, and just the other type of foliar structure is the spade. So in the whole inflorescence, we only have the spade, which is similar, which is basically larger than the bracts, and that's only the two types of morphology that we can find in foliar structure found in the inflorescence.
Thank you.
Are you planning, have you published this? Do you plan to make it available? I find it very interesting because in Epidendrum we have basically the same thing. But it would be very useful to have a copy of your definitions and descriptions.
Yeah,
illustrations.
this work is currently in press, so maybe in 1 month or 2 months it's going to be published. No, in this journal called the Botanical Review. Any other question?
Have you looked at the microscopic, more like flower primordium?
No,
of
no.
These?
What
would you expect if you're going to look at?
Sorry?
No, what would you expect? Like, would you expect a lot of diversity in there? But I wouldn't expect much. I don't know. What do you think?
I actually don't know what to say about this group. So, since we have a clear typology, I think that is not so necessary. I don't know.
Thank you very much.
Thank you.